is Eileen Furlong. She's a molecular biologist working on the regulation of the genome. She is head of the Department of Genome Biology at the European Molecular Biology Laboratory in Heidelberg. She received her PhD at the Conway Institute at the University of California, Davis, and her paper is titled Understanding the Impact of Developmental Perturbations with a New Level of Resolution.
Great, thank you so much. Yes, I'm really sorry not to be there. I first want to thank Uta and Ellen for organizing the meeting. Again, I apologize not to be there. I had some on Friday lunch with some people that tested positive for COVID, and I had to say I chickened out. I was afraid to risk it. Yeah, but also I'm particularly honored and excited to be here, and I'm sorry I'm missing the conversation for this meeting in honor of Eric Davison, who is a big hero of mine in terms of developmental gene regulatory networks. And I guess I just heard from the last speaker, there's probably lots of conversations at the meeting about this sort of interplay between how do you encode, I should say, like plasticity on the one hand versus all of this diversity during embryogenesis on the other. And as Eric really pioneered this field of thinking about gene regulatory networks and how we start to engineer and think about these almost as an engineering problem, and how could we digitally encode these. And I think at the time, over the years, lots of us have been trying to think how can we move this, including Eric, to capture all of the dynamics and the continuums within this, which these kind of representations do happen very well. So I think we now, in my opinion, have the tools to really do that, and that is within single-cell genomics. So what we'd like to do, as I said, is to be able to capture developmental networks or the molecular biology that's underneath these, all of the dynamics and the continuum of that. And as I said, I think genomics is giving us, and microscopy of course, great chances to do that. And what I'm going to tell you about today is how we're starting to use single-cell, what I call regulatory genomics, and that is using a method called ATAC-seq, which is a method that allows you to identify all of the regions in the genome that are occupied in one go. And this allows you to identify, for example, dynamics of enhancer occupancy, which are really the sort of regulatory elements or switch elements within these gene regulatory networks. You can think of these as the nodes within the networks. And we showed in a very nice collaboration with the Shendor lab that you can first of all separate cells. So this is single cell data. And you can separate each dot here as an individual cell. This was just a whole embryo, and now doing one time point, it's Drosophila, single cell ATAC-seq, and you can see that all of these cells separate into nice clusters, and we could annotate what they were based on the common usage of the non-coding genome. So this is basically of enhancers, or putative enhancers, and actually we could show, I'm not going into the details of this, but we could show in transgenic embryos that the vast majority of these regions are enhancers, and that when we test them, for example, they're active in the cell type that we expect. So this is just important for you to kind of think of in the back of your mind, that when I'm talking about these trajectories that I'm now going to show you with single-cell ATAC-seq data, that lots of this is really looking at the usage of developmental enhancers through time. And what I want to kind of discuss is a little bit of how, thinking of this plasticity in developmental networks, how can we capture this plasticity, and how can we move beyond the sort of field of capturing single cell sort of atlases. And I think for that we need to perturb the system in order to be able to really model it. And we also need very high temporal resolution. We need sort of a continuous trajectory, because we can learn a lot from what happened in the cell before to what's going to happen in the cell in the future. And I'm going to sort of touch on these two today, hopefully. So first of all, for the perturbations, we've been doing that in two different ways. So one is perturbing systems in trans, so mutants of the transcription factors. Also newer optogenetic depletion, which I'm very excited about, and also perturbations in cis, which I won't have time to tell you about. So first of all, for the perturbations in trans, well, as I mentioned, what we want to try to do is get to the point to see how far can we now really push the system to gain the data where we could eventually model a tissue's development. or in our case, we're even picking a germ layers development, and that is the mesoderm, which is specified into different muscle primordia, in different muscle tissues. And for this, we're using Drosophila as a model system, because this mesoderm specification occurs actually in all model organisms. But we're using this because, on the one hand, it's obviously simpler than humans, there's less cell types, less transcription factors. But on the other hand, it's also incredibly complex. I mean, the end of embryogenesis, Drosophila has a beating heart, a nervous system, a brain, it can crawl, it can digest, etc. But as I said, the ultimate goal would be to see to what level of experimental data do we need to be able to sort of model these transitions, and also to what scale do you want to do that at. But for me personally, I'd like to be able to predict the effect of mutations, for example, within that trajectory. Okay, so to do that as a starting point, we do have to build an atlas, or we did, and for that, Stefano, which is just an amazing PhD student, collected stage 2-hour collections. This is a nice thing in Drosophila, you can really get 2-hour collections, stage collection, sorry. So we did this for overlapping 2-hour time points, hoping that we'd be really able to get a continuous view. And for each 2-hour time point, we fax purified out the mesodermal cells as they were being specified along this trajectory. And so when we do that for all of these time points, together we have about 25,000 cells that we did single cell ATAC-seq on. And now I'm just going to show you what happens if you just cluster all of those together. So when you do that, you can see that you naturally get this tree-like structure that's formed. Actually, there should be a branch here. And I'm color coding that just based on the colors of the collections. So here you can see that time just naturally falls out of this. Again, each dot here is an individual cell. And now this is just based on single cell ATAC data, so it's sort of which elements of the non-coding genome and promoters are occupied in all of those cells. And you can see the first axis here is time, where the immature mesoderm is down here, it's unspecified. And then you can see there's lots of branching here. And actually this is very exciting to us because this is the stage where it's known from decades of Drosophila research that the mesoderm starts to be specified. And we can annotate what these cells are. I won't go into details of that, but we can tell what's the visceral muscle, the gut muscle, the heart, the body wall muscle, and so on. And from this type of molecular data, you can also zoom in. I won't go into details with this, but you can zoom in and identify, on the one hand, which genes are changing along a given trajectory. You can say, okay, let me look at, from the unspecified mesoderm to the visceral muscle, give me all the genes along there that are changing, and give me all of the regulatory elements, these putative developmental enhancers. So the huge wealth of data describing the system, let's say. But you can say again, this is still the sort of atlas building. So how can we now go beyond that to start to see, can we perturb the system, and can we start learning the input of each of these transcription factors on that system? And these factors are known, as I said, from decades of genetics work. And for that, I'm going to show you how we've been doing that first with loss of function mutants. And before showing you that, I just want to remind you a little bit of basic genetics. But I know this is a very interdisciplinary audience. So if you think of a recessive lethal mutation, like a loss of function mutant, normally that will have to be maintained over a wild-type chromosome for that gene. So then the males and females of that, only 25% are your homozygous mutant of interest, 50% will be the mutant over the wild-type chromosome, and 25% wild-type over wild-type. So if you work, for example, in mice or in fish, you would have to go down and, for mice, for example, T-tail and find out which are your mutant embryos of interest and separate those and do your experiment on those. What we wanted to first see is now, could we define a way where we could very rapidly just collect all of these embryos, and then after the experiment, digitally genotype each of the embryos, each of the nuclei. So for that, we've developed what we call sort of a digitally genotyping nuclei algorithm. So what we do is we collect pools of embryos of mixed genotypes. We basically pop out the nuclei from all of them, perform single cell ATAC-seq with all of them, and then afterwards, because we're actually looking at the DNA here with the single cell ATAC-seq, we can then digitally, computationally genotype each nucleus and put them into the 3 different bins here. Are you a homozygous, a heterozygous, or a homozygous wild type, for example? And we could do that very well. I'm not going to show you the data for that, but just believe me, I'm happy to discuss it afterwards. We could do that like with over 99.9% accuracy, and we could prove that by hand sorting the mutant embryos as well. So then let's go back to this, and what does that mean then? So that means that we can now just collect mutants from the parents and very rapidly profile those. And we have done that for 4 different mutants in this transcriptional cascade. And I'm just going to show you the data. So these are the 4 mutants here, the data from those. So this Tinman is NKX 2.5, bagpipe and U. For each of these factors, then, as I said, we took all of these embryos, we performed single cell attack-seq, and then we integrated all of that data together with the wild-type trajectory. And so the key thing is here having a very nice wild-type trajectory. And when you do that, this is what the data looks like. Okay, the clustering or the presentation here is a bit different, but this is the unspecified mesoderm, the visceral muscle, the heart, body wall muscle. You can see from all of these cells. So now we're up to over 40,000 single cells that we have data for. And actually having these heterozygous nuclei or embryos in there is very good because it allows us to, it's technically just good to integrate all of the data together. But let's get to the mutants then. So what happens then when you start looking at the homozygous mutants? The question is here, can we phenotype those mutants de novo? And that's what I'm going to show you or hope to convince you of. When you first of all look at the heterozygous nuclei, so these would be heterozygous embryos, you can see that the cells map into the heart here or the visceral muscle. And actually these are the exact tissues that this transcription factor is expressed in. And now if we look at the digitally genotyped homozygous nuclei, the first thing you can see is that we don't get 25% as expected, we get less than that, we get around 16%. So where do those cells fall on this trajectory? Well, when we look at that, we can see that first of all, we have absolutely no cells in the heart, and we have some homozygous cells in the early mesoderm. Sorry, this is going from this direction to differentiated is here, these are the older. So we see that we have visceral muscle cells being specified, but they're not differentiating into the visceral muscle, and we have no heart. So the prediction from this would be that these embryos would have a heart defect and a visceral muscle defect. And that's exactly what you see. So this is a Drosophila embryo here, stained with a muscle marker, and this is actually a bit difficult to see here, I can see, but this is the little Drosophila heart here. And if you zoom in, you can see the gut musculature. And now if you look at a homozygous embryo, there's absolutely no heart, and there's no musculature at these late stages of embryogenesis. So this was very exciting to us because it meant that just by performing this molecular experiment together with this tight developmental time course where you can map out the trajectories, we can now phenotype that mutant without looking at a microscope. But I think what's really exciting about it is that you can also quantify that phenotype. So for example, we can now say exactly how many cells, what fraction of cells are missing from the visceral muscle, and at exactly what stage of visceral muscle development. And you can see this here a little bit when I'm showing here. These are the different clusters. It's a little bit technical. And there's different clusters along this trajectory here. And what's showing here is depletion. We can see exactly when we get depletion and what is the fraction of depleted cells within those. So these are the cell numbers going down. So of course one could get this from the microscope, but it would be actually very time consuming. But the other exciting thing is we could see that we got a statistically significant gain of cells in other muscle lineages, which I'm showing here. So it's small numbers, but it is highly significant. So this is again getting to this plasticity. So although now in this phenotype we have actually a number of things going on, whereas classically you would just think, well, Tinman, you end up having no heart or visceral muscle. But that's an oversimplification. Actually, there is no heart. The visceral muscle actually is specified, it just doesn't differentiate. And that a small fraction of these cells can take on other mesodermal lineages. And that's something that wasn't shown before. And I think this is now a for us to start mapping on phenotypes or getting to phenotypes, for example, that we may not have sort of seen before. I'll just show you that for one other, or two other examples. Actually, these 2 factors here, bagpipe and Ben-Gurion. Again, these 2 factors are specific for the visceral muscle here, not the heart. So you can see in the heterozygous situation, Again, when we look at the homozygous situation, we don't get 25% of the nuclei, it's again lower. So this is also suggesting that there's some cells are missing here. And that's exactly what we can see here. We can see that the visceral muscle is specified, but we don't get late stage visceral muscle here. And that's exactly what you see in the mutant. So this is a heterozygous situation. Now we're zooming in, this drosophil embryo zooming into the visceral muscle here. In the homozygous embryos, you have absolutely no visceral muscle specification, or visceral muscle, I should say, differentiated at this stage. So this is again showing that you can de novo phenotype these mutants, and as I said, it's a very quick way now where we can just take all embryos from that litter and just quickly do these experiments in one go. Now what about this other mutant? I'm going to show you a different type of phenotype. So in this mutant, we actually did find 25%, and the corrected percentage. So this was already telling us actually that we don't have a loss of cells, which is interesting. When we cluster these with the trajectory, I'm sorry, this is just a technical thing, but it's the unspecified mesoderm, here's a visceral muscle, it just looks a little bit different. This is going in developmental time in this direction. So these are the older cells, the older embryos. So you can see then, well, where are these homozygous mutants cells? And when we look at that, actually they are falling off the trajectory. So in this case then, the cells are still specified in the homozygous mutant. This is myocyte enhancing factor. But they are not on the wild type trajectory. So you could say, well, this is our wild type trajectory here is mesoderm being specified into different muscle. So you could argue, well, maybe these cells don't know that they're mesoderm. Maybe they're forming some other tissue, even in another germ layer. And we could actually test that because we also have whole embryo single cell attack data of all lineages. So we could then just simply cluster the molecular profile of these mutant cells with all other cells. And when we did that, we could see these two mutant cells are here, and they're clustering more similarly with muscle cells, and specifically the somatic muscle cells, very far away, for example, than other tissues from other germ layers like the hindgut, endoderm here, the central nervous system, and so on. So it seems that these cells do know that they're mesoderm or that they're muscle. And actually what's also really interesting is that they're not only know that, but they're not just blocked in an earlier mesodermal stage. They're actually progressing in their development to the right stage of this trajectory, but they're just off the trajectory. So this is suggesting that they're somehow in some alternative state. So the nice thing about these experiments, well, in my opinion, is that it doesn't just give you then this cellular information of what's the state, but also obviously the molecular information. So we can now go in and see what's going on with these cells. So we could see with these MEF2 mutants, which open chromatin regions, which enhancers or promoters now are have lost, like are gone down in their openness, and the proxy for their binding, and which ones have gone up. And you can see that the majority of them have lost or are closing, have lost their occupancy. And this is absolutely to be expected. So this is myocyte enhancing factor, regulates a lot of contractile proteins. So this is myosin light chain, for example. And in the heterozygous mutant, you can see that known enhancers here are open. Then the homozygous MEF2 mutant cells, these are from the single cell data, are closed. But we also have these ones that are gained. And when you look at those, actually it's very interesting. You can see, for example, this is a neuronal enhancer, actually curated from Alex Dart's lab from the Vienna tiles. And you can see that in the wild type trajectory, this is absolutely closed, so in the muscle and the mesoderm. But now in the MEF2 mutant, this is open or occupied. So this is suggesting now that these cells on this trajectory are in this sort of mixed cell state where they have both a loss of muscle, molecular, like muscle enhancers, let's say, activity, and a gain of inappropriate neuronal, and actually we also see some ectodermal ones as well. So just to kind of summarize from this part, what I hope what I'm excited about and I hope to convince you of is that by applying a very dense developmental time course of your tissue, you can use that information then when you combine that with mutants to gain information of what I call sort of cellular phenotyping. You can do this sort of de novo where you can see, okay, the late visceral muscle, etc., is missing in that mutant. And what's exciting about this is that we can also quantify these phenotypes and even identify new, more subtle phenotypes that weren't seen before. And also, on the other hand, you can molecular phenotype these mutants all within the same experiment. So you get the two of these, well, for the price of one, as it were. Okay, so now we are continuing with that. Obviously, we're quite excited by this, but we're also, instead of normal loss of function mutants, want to apply another perturbation. And the reason for that is oftentimes, if you go back to something like this, when you have a developmental mutant, for example, something like twist or MyoD, in a classic loss of function mutant, the development of the whole trajectory can be blocked earlier, so you can't actually see what's the role of that transcription factor later. So you need some way of doing conditional perturbation later in embryogenesis. And for that, we have been developing this optogenetic depletion system, which I just want to describe now. Because actually, you guys, well, anybody could use this in your system. So this is based on Lexi. So this was a method that was first of all developed in tissue culture by another group in mammalian tissue culture. It's based on having a love domain with a nuclear export signal. So you basically tag your protein of interest with this domain, you shine blue light on it, and it causes a confirmation on unfolding, then the nuclear export signal is exposed and your protein can be exported from the nucleus. In our study, we made a whole series of mutations to the love domain to try to increase its strength, and we did increase its strength, and we changed its dynamics of its recovery. So we call the system iLexi, and I'm just going to show you a little bit what that looks like. This is actually a Drosophila embryo type of twist. Actually, that was so fast you probably didn't see it. This is twisted in the dark with the blue light on. You can see that here when the blue light goes on, boom, you can see it's already going out of the nucleus. This is actually so rapid that this scale here is in minutes. This is, not like a Negev system which can take, people do this for hours or 24 hours, etc. Here you have the nuclear cytoplasmic ratio. Put the blue light on, the factor's gone out. We turn the blue light off, the factor starts to return. And we, as I said, we have different systems which have different recovery dynamics. Because this was almost too fast actually for some experiments. You can see here that it's recovering. So you can use this system very nicely then to look at things, even within an individual cell under the microscope, because you can, target your blue light depletion to very discrete parts of the embryo. But also for biochemistry or for genomics experiments, we want to do this with quite a large number of embryos. And for that, we've developed these blue light LED boxes. So these LEDs are super cheap, as you know. So we have just a nice system where we have blue light LEDs in the top and the bottom. Put your embryos here, and then you can zap them from both sides. We can control the pulsing of the blue light and the duration. And that works extremely efficiently. So we're now going through and doing that with each of these different factors. So now you can imagine that we can start to take them out, put them in at different stages of embryogenesis and start looking at their contribution at each of those stages. So I don't have the data for that, but I just, this is where we're going. But now I want to just in the last part of my talk sort of switch gears a little bit, and you can think of this as sort of zooming into the nodes of gene regulatory networks. As I said, these are really the enhancers to get back to how do they work. And I want to just discuss this with you because it's a little bit maybe provocative at the end. I want to hear what's your opinion about this. I've only presented this, I think, once before, and also if you have any ideas of how we could actually test this hypothesis. But basically, well, first of all, our question originally was trying to understand how do enhancers communicate with promoters. So if you think of the target genes, so genes, as a number of people, I'm sure Alex and others, and Denny Bull have discussed it, but genes have many enhancers. They can be located 5' or 3', and even in the intron. And the key question is now, why does this enhancer regulate, this gene and not this gene, and so on. And we know, though, that they come together to regulate that gene in some kind of 3D topology, 3D loop, which is also something that many of us are trying to understand how it is regulated in terms of developmental time. And actually, much of what we know about this has been obtained from very careful work on model loci, originally particularly the alpha and beta globin loci, but also the HOXD cluster from Denis and Sonic Hedgehog locus from others. But from these two particular loci, which are actually unusual. So these are clusters of genes regulated by a cluster of enhancers, a locus control region. And what they found was that, these were very early studies looking at chromatin topology, was that in an inactive state, or actually not in the erythrocytes, the cells that the genes are active in, that the enhancers were very far away from the gene of interest, and that the enhancer only came in proximity at the time when the gene was expressed. And this has led to the model that it's actually the proximity alone of the enhancer and the promoter was the trigger for gene expression. And I should say that Jared Global's group has shown for the beta globin locus that if you force a loop, this is sufficient to trigger gene expression. And this has actually also been shown now in Drosophila as well by Thomas Gregor's group. So we wanted to see to what extent does this hold true for like normal enhancers, let's say, not these clusters of enhancers. And this I'm going to go through super quickly because I've discussed this many times and it's old at this point. But it's important for what I'm going to say next. So we just took different stages of embryogenesis, mainly this stage here. So this is the stage of specification. This is actually the point where I was showing you in that tree where you get lots of branches because you start to get cell-phase specification here. So we took 100 developmental enhancers and looked at the proximity of those enhancers in two stages of embryogenesis. The stage where they were off, and the target genes were off, and the stage where they were on. Basically we were expecting to see lots of dynamic changes, etc. So this early stage is really at the blastoderm stage. But to make a long story short, we didn't see that. We saw that the enhancers in proximity at the time they were active. This is what you'd expect. But when we looked earlier, the surprise was that they were already in proximity at its earlier stage. This is about 4 hours earlier. So you could argue, well, maybe they were in proximity then because Drosophila embryogenesis is very quick and very dynamic. As I said, typically people look over days at ES cell differentiation. Here we're really talking about hours. But that's actually not the case. There's been a number of other studies which have found a similar thing in different systems, in vertebrates as well. So there seems to be, I would say, two types perhaps of loci, or two types of, conditions of some where the enhancers seem to be far away from the promoter when the gene is inactive, or in cell types where it's not active and it only comes in proximity at the right time, and other ones where it's already in proximity before activation. And in our case at least, we saw that these promoters have evidence of Pol2 pausing, so it suggests that everything is primed earlier and ready for activation. And actually what's interesting is that another study, well, these are just some of what I have just said, that actually this I think is conceptually important because it would suggest that at least in these cases that enhancer and promoter proximity, at least at the level of what we can measure with chromatin capture and also with light microscopy, is not the trigger for gene expression in some cases. Whereas I said it appears to be in other cases where you really force a loop, and it is. Okay, and as I said, there was another interesting study that's just been published, recently published in Drosophila, I guess last year, which is also showing that if you go back at this earlier stage, as I said, the enhancers are in proximity here, they're in proximity here. If you go back even earlier, they're also even in proximity here, even at the stages where before TADs are formed. This was at least looking at two enhancers and promoters by imaging. So what I want to kind of discuss now is what are we doing now, where are we going from here? So we started to also go later in embryogenesis. As I said, this was comparing now a blastoderm stage, really pluripotent stage, naive stage, let's say, to cell-phase specification. And we've now gone later in embryogenesis. So what do we observe? As I said, so that's what we compared before. We're going later, and again, you could think, well, it's only 2 hours later, 2 to 4 hours later. But at this stage already, you have terminal tissue differentiation. As I said, things are moving extremely quickly here. So what was the experiment that we did? Well, we fax purified nuclei from 2 different tissues. And we did this with this method that we call BITS. I mean, it's just, in case any of you want to use it, it just stands for batch tissue specific. But the whole point of the protocol is that you fix your tissue complex sample. Actually, we have colleagues that have used this in zebrafish, also in mouse brain, neuronal tissue. The trick of the whole protocol is that you then pop out intact fixed nuclei. We can then stain these nuclei and then form FACS on nuclei, so it's actually, well, FANs really, fluorescent activated nuclear sorting. And then you can use those nuclei to do a whole host of different, genomics experiments, let's say, in a tissue-specific way. We're now doing sort of 3-way nuclear sorts using antibodies from the same pool of embryos. So in this experiment, what do we do then, to go back here? So from these now 2 time points, we're going to compare this with this. We FACS purified out the nuclei from neuroblasts at this stage, are specified to be neuroblasts and differentiated neurons, and also myoblasts and differentiated muscle. We then had those very pure populations of nuclei, and from each of those then we performed capture-seq. This is one of these chromatin conformation capture methods, which allows you to go from one region of the genome, let's say an enhancer, to see what is in proximity to genome 1. So we did that Tim here hand-picked, went through the literature and selected 100 embryonic enhancers. No, sorry, that's previous. So 300 embryonic enhancers that have really tissue-specific activity. So they're either in the muscle or in the neuroblasts. And similarly, we had 300 tissue-specific genes, either expressed in neuroblasts or in neurons or in muscle. And then for each of those, then you could then see, okay, what is proximity? So this is just showing the dynamics of all those. We have these that are muscles, and we have ones that are on, going to off, going to on, etc. But we have lots of specificity and tissue-specific dynamics here with what's called your baits, the region of the genome you're looking at. But then you can then see with these muscle-specific enhancers, what is your topology then? How are you looping to your promoter between myoblast now and terminal differentiation in muscle cells? But also with those muscle-specific enhancers, what are they doing now in neuroblasts and in differentiated neurons? And vice versa for the neuronal enhancers in the muscle cells, as well as in the neurons. So for all of that then, we had a ton of data. We complemented that with some other data, but just to go into this data. So you could say, first of all, well, what did we see? Well, we saw on the first hand, you could say, similar maybe to what we saw before, that most interactions don't change, even though the activity of the bait does change. So this is looking at the interaction. So we had a very similar median here. If we now just are looking at enhancers or genes between the 2 states that are going from off to on or on to off, or even if you compare on to on versus off to off, so the ones that are just not on, etc. So that's the first thing, it's just to say that the majority are within this window. But there are quite a few regions that are changing. So let's look at those regions that are changing and see when are they changing, do the changes make sense, etc. And that's what I'm going to talk about for the rest of the talk. So what I'm showing you here is just sort of a cluster of those changes. So if you just think of this heat map, then we can see at what stages of embryogenesis are things changing. So we can look now in the mesoderms, we can think of this as myoblast versus differentiated muscle, neuroblast versus differentiated neurons. And we can see that if we clustered all these changes, mainly what we're seeing is that most of the changes here are happening at the second time point, during this terminal differentiation, not during the self-date specification. So let's see now, well then you can also ask, well, do they make sense? So do the changes that are in the differentiated neuron, are they enriched for the baits that were, or the differentiated muscle, the baits that were active in the muscle, and they are, and vice versa, the ones where we see interactions now, differentiated nervous system are only at enhancers and promoters in the nervous system. So actually, it seems that at this late stage, there's a lot more things happening than at these 2, these comparisons here. So let's look at that now in another way. So now we have asked, with this, I can think of this chromatin topology information, if we just now cluster all cells together and see how close are you to each other, what we can see is that neuroblasts and myoblasts, sorry, this should be labeled myoblasts, are actually much closer than they are to each other. So in terms of their 3-dimensional enhancer-promoter interactions, there's hardly any difference between those in a neuroblast and in a muscle compared to each other, compared to, for example, a neuroblast and a differentiated neuron. So this, I think, is really amazing. You think of this as a dendrogram. The branch length here really represents here, well, a good framework to like quantitatively and systematically look at this. So let's show you a couple of examples of this. So this is what this type of data looks like. So here you're looking now at your viewpoint, your anchor. This is a promoter of a neuronal gene. You can see that if we look at this, these two time points here in the earlier time point of specification, there's not much changes here. But then if you start to look at the differentiated muscle, you start to get specific regions of interaction and differentiated neurons here. Similarly for this gene here, zinc finger homeodomain, it's a gene that's expressed in both of these two tissues, and you can see specifically in the differentiated, terminally differentiated state, we can start to see new interactions, and not in the earlier state. So what this is suggesting, at least here, is that, well, the interesting thing I should also say is that we also see very little loss of interactions at this later time point. These new interactions all seem to be gains on top of what we're calling sort of a pre-existing landscape. So let me just go back here, and this is where I'm being a little bit, well, wondering what you think, or being a bit provocative when we think of this, because thinking of sort of one of the questions of this meeting of developmental plasticity, you could think when you think of sort of this, you know, overused Waddington landscape, but when we look at these stages of very early on, the blastoderm stage when things are not specified at all, to specification, in terms of topology of enhancers and promoters, this is very similar down different lineages, all at these stages here. But now when we look towards this endpoint now, when things are really terminally differentiated, we see a lot more differences. So we're just wondering, or the provocative question here is, you know, does the similarity up here then sort of facilitate and allow for things like transdifferentiation to occur in embryogenesis at this stage, whereas this changes in topology down here would really help to sort of cause this developmental lockdown, which is something actually that Eric has proposed a long time ago in terms of the structure within gene regulatory networks. But is this structure of the chromatin topology on top of that another way that could facilitate that lockdown for terminal differentiation? Sorry. And with that, I think I'm going to stop here. I'm very happy to take questions. I should just, first of all, though, thank the people in the lab that did the work. The first study was done by Stefano, which is an amazing PhD student. And then all of the chromatin topology work was done by Tim Pollock, who's here. And he was joined by Adam, who did the computational analysis. And with that, I'm happy to take questions.
Yes.
Eileen, can you hear me? Dennis?
I can hear you, yeah.
Okay.
Hi, Jenny.
Eileen, regarding the comparison you made between the globin system and no contact, or the sort of pre-contact that you see in the system, do you think that is due to the fact that in the globin system you get super enhancer, which at some point has to shift from one promoter to the other to make the the globin switch, in fact? So do you think that you do not get this constitutive contact before, because that would be too risky if you think that you will have to shift? So the question is, do you think that these differences you will find the same whenever you will look for genes which are at the very end of the differentiation pathways, where you need a huge activation of a single promoter, I would like a keratin or whatever, which would have probably a high probability to have a super enhancer?
Well, I mean, I could only hear half the question, by the way, but I think I got the question. So it's, I think, do I think, what I heard was, do I think that this is really genes that are sort of at the end of the pathway, sort of terminal differentiation genes? I think you mentioned keratin or something like that. I mean, I think that these really are those terminal, like genes that are expressed in terminal differentiation. But they're not all effector proteins. Like some of these are also transcription factors. In terms of, do I think this is a switch of a promoter and a super enhancer interaction? Was that what you asked?
No, I meant the globin, they have to switch, from a fetal form, an embryonic form, a fetal form, an adult form. So if you would have strong pre-established contact or proximity, perhaps that would be detrimental or the flexibility of the switch.
Absolutely, absolutely. Yeah, I totally agree. Exactly. So that's what I meant. I totally agree. So, you know, that's what I meant also, like if you want to do any, well, transdifferentiation is probably too, like maybe a rarer case of that type of plasticity, but absolutely. Yeah, I agree. I mean, we don't have, well, by the definition of super enhancers being a cluster of enhancers in 12 kb, I would say Drosophila is a super enhancer. So that's you know, we have clusters all over the place, but I totally agree. I think it would potentially allow for this plasticity. At least for me, it's very telling that this is we have these very little changes in these preformed topology. Everything is in proximity from the early to this cell-plate specification, but then we see a gain on top of that at this later stages where things really are terminally irreversible, like terminally differentiated.
We have time for a quick question.
Sorry.
Eileen, it's James.
Hi, James.
So I'm choosing my question. So I asked the MEF2 question. So I was struck by that result where in the MEF2 mutant, the cells are adopting some aspects of a neural-like gene expression profile or identity. And that sort of reminded me of Marius's Vernig's experiment reprogramming fibroblasts with neurogenin 2, where there's a side population that he gets in those experiments which are actually muscle-like. And his interpretation of that is that the neurogenin 2 is taking on a MyoD-like activity, as they're both E-box proteins. And that, I was wondering that, so is MEF2 somehow ensuring the specificity of, what is it in Drosophila, so a MyoD equivalent in flies?
So is MEF2 at the MyoD equivalent in flies?
No. Is MEF2 ensuring the muscle specificity of the MyoD equivalent?
No, well, I mean, there is a MyoD, well, I think twist is a factor that seems to be doing the same as, and actually twist regulates MEF2's expression, so MEF2 is down upstream of that. And originally MEF2 was always described as a differentiation factor, but actually if you go back and check the old papers, there is very low, and we've checked that in embryos as well, there is a low penetrance phenotype where you actually get a loss of some muscles as well. So it is playing an earlier role. But I think your analogy though is quite a good one. Well, it's an interesting point that we've been thinking about because, you know, MEF2 is also involved in vertebrates with neuronal development with all of the different isoforms. And there is MEF2 also in Drosophila brain as well. So it could be some, like it's not in the embryonic brain, but in the adult. So it could be something like that where these, you know, the factor can, in different contexts, bind to these enhancers, but it shouldn't be able to regulate them now, and somehow we're seeing this aberrant effect, or it could be an indirect effect. This we can't, you know, it's difficult to rule out. But we just think it's I should also say it's not specific to the nervous system. We also see some ectodermal, and these are really at the enhancer level, like the open chromatin level regions coming up as well. So it seems like they're just, yeah, we're sort of calling it a mixed cell state. They don't really know what they are.
Thank you.
Thank you.